Hi, everybody. Nice to be with you again. Today, I'm going to tell you a little bit about some information that will be really useful for you to know in advance of reading our paper on limb loss in snakes. One important concept in genetics and evolution and development is the concept of an enhancer. An enhancer is a particular DNA sequence that a transcription factor, which is a protein, binds to. So in this example, this cartoon shows an enhancer sequence, a particular sequence of DNA nucleotides that a particular transcription factor in this case will bind to. So here they've described two different enhancer sequences that bind two different proteins. And enhancers act to enforce or to support transcription by, as shown here, sort of stabilizing or holding other proteins in place that are necessary for transcription, like RNA polymerase and other associated transcription factors. So enhancers are not essential, but they're useful for promoting transcription of a gene to cause gene expression. So enhancers are different from the promoter because the promoter is where the general transcription factors bind. And the promoter is usually just next to or just upstream of the transcription start site, what we typically think of as a gene. So those proteins all recognize specific DNA sequences in the promoter. One of the most well-known is the TATA box, a sequence of TA that TATA binding protein binds to. And so, for example, I think that protein might be TBP. It's a general transcription factor. TBP stands for TATA binding protein. So it binds and recognizes that DNA sequence. So TBP in itself is the first transcription factor that will bind a promoter to start the process of transcription. And it then recruits or binds to and holds on to these other transcription factors, and the whole complex then grabs, essentially, and holds RNA polymerase at the promoter long enough for RNA polymerase to start transcribing the DNA and producing an RNA transcript. So that is what an enhancer is. It's a DNA sequence that a transcription factor will bind to. And these transcription factors that I'm talking about, the ones that are cartooned here as binding enhancers, these are not general transcription factors. Again, general transcription factors are ones that are used every time RNA polymerase starts transcription, anywhere in the nucleus at every gene. So the way the cell controls which genes are transcribed are not through regulating the general transcription factors, because they're always used every time there's transcription. Each individual cell then controls which genes are turned on and turned off by producing different combinations of transcription factors. So for example, if in this cell type, this gene that produces that transcription factor is not being transcribed itself, and that protein isn't present, then there will be no transcription factor to bind this enhancer. And the loss of that binding, because this protein isn't being expressed, could be enough to not allow RNA polymerase to bind to this promoter and to start transcription of whatever this downstream gene is. So the combination of proteins, transcription factors, differ from cell to cell and over time too, within the same cell. And it's these combinations of transcription factors that essentially make a code for the cell in terms of which proteins are turned on or which genes are being transcribed at a particular point in time. And so that concept is made in this slide, which simply shows that there are combinations of enhancers that specify different types of cells. So in this case, from a basic tissue, the production of PDX1 without ALB transcription factors causes development of pancreas tissue, but there are different types of pancreas tissue. So those tissues then that also express the transcription factor P48 turn into exocrine cells. And instead, if they express this set of transcription factors, they become endocrine cells. And then each of those different types of endocrine cells has a different, again, code, essentially, of which transcription factors are turned on Those again go on and turn on different sets of downstream protein or genes, regulate their transcription. And that can help identify individual cell types within a larger tissue. A really important concept in gene expression regulation is the difference between cis regulation and trans regulation. And this is sometimes a difficult concept to understand, so I'm going to explain it briefly now, and hopefully we'll have time to discuss this more in detail as we go. So you may be familiar with the idea that the prefix cis means same or together, and trans often means across, like transatlantic. A boat or an airplane that crosses the Atlantic Ocean is a transatlantic airline or boat. So there are types of mutations and regulation that are classified as either cis or trans. So in this case, let's imagine that we have not only this part of the chromosome, but another part of the chromosome that also has, I don't know what color that is, red or green, the enhancer sequence. I'm going to assume it's red. I'm colorblind, so don't hate me. So there's another enhancer. And there's another gene that it regulates. So we usually use the right-angled arrow to indicate the transcription start site of a gene. So here we have two different genes, one here, one here. We have two different enhancer sequences that will bind the same red blobby transcription factor protein cartoon here. And it's important to note that any transcription factor, be that a general transcription factor or one of these specific transcription factors that binds an enhancer, will have a part of its surface, the shape and the structure of the protein, that is a DNA binding domain. That is, those amino acids are in a particular shape when the three-dimensional structure of the protein folds. And that shape is specific, targets that protein to a specific DNA sequence. In other words, the proteins, as you might expect by now, can recognize different DNA sequences. And these transcription factors bind to specific, although sometimes not completely specific, but generally specific, if that makes sense. There's a little ambiguity. Sequences. So for example, it could be that this protein will recognize, I'm going to make up a short enhancer sequence, AT-AT. And this other enhancer sequence might have AT-AA. So they're similar in sequence, there's a slight difference. So maybe this same transcription factor would bind this DNA sequence very strongly because it prefers, in this case, I'm making up that it prefers AT-AT. That same protein might still bind the enhancer sequence AT-AA, maybe just not as well. So maybe you wouldn't get as much transcription from that gene. So DNA sequence differences in the enhancers can influence transcription. And that's an example of cis regulation. This DNA sequence change, which I'm going to put an asterisk on, that fourth nucleotide is an A. And that confers upon this gene, in my example, reduced expression. It will probably produce, maybe produce, a little bit less transcript than this transcript because it has the consensus or the perfect sequence that that transcription factor protein recognizes. So that is an example of a cis regulatory effect because, and this is the cis part, this mutation only influences the expression of the gene that it's cis or together or near to on the chromosome. In other words, this mutation doesn't influence the expression of this gene, which uses the same transcription factor, because this element, the enhancer, is what we call in cis, or together with, the gene that it regulates. So this mutation doesn't influence other genes that are farther away from it or on different chromosomes, for example. So this is an example of a cis mutation. Now, let's consider a transmutation. So further, elsewhere on that chromosome is the coding region of the red transcription factor. Right? It's a protein, which means it's encoded by a gene, and that gene has to be somewhere on the chromosome. So here's the red transcription factor gene. Let's say that we have a mutation in the red transcription factor DNA sequence of this gene that produces this protein. So now consider this is a new scenario, not related to what we just discussed. Let's imagine that this mutation happened in a particular cell. And so after transcription and translation, that produces a protein transcription factor with a slightly different shape. So I'm showing here, instead of being totally round, it's got a flat surface. And what I'm trying to indicate by that is that it could be that this mutation that happened in the gene causes the protein, the transcription factor, to fold differently, to adopt a different three-dimensional shape. And it could be, and this does happen, but in this example, it could be that that mutation makes the transcription factor no longer have a DNA binding domain. So here's where the transregulation comes into effect. How is this mutation going to affect transcription in the cell? Well, if this is the only gene that produces this transcription factor and it's mutated, then that protein will not bind to ATAT, the enhancer sequence upstream of this gene, and it will also not bind to that enhancer, the ATAA, which in the previous story, it didn't bind as well to, but now it doesn't bind at all. So this one mutation has had a trans effect because it doesn't just affect the expression or the action of genes that are near it on the chromosome, which would be in cis. It produces a diffusible factor, and that's sort of the key to transregulation, is the mutation causes an effect that has a broader reach. This protein now influences the transcription of all of the genes on the same chromosome, on different chromosomes in the same nucleus. And so that's why this is a trans effect. So cis mutation or cis regulation has to do with nucleotide differences or mutations or sequences that have effects only nearby physically, nearby on the same chromosome usually. Trans effects are effects that usually are involved more in mutations that affect proteins. And those proteins are diffusible in the nucleus and in the cell. And so those can have effects on multiple genes or multiple loci. So that's the difference between cis regulation and trans regulation. There's one type of plot that a lot of evolutionary and developmental biology papers are going to contain, and that's a VISTA plot. And so I'm going to show you exactly what this looks like as an example. So here we are at the VISTA website, and we can look, for example, at a pre-computed alignment. So we're going to look at one alignment right now. We can load, let's just pick a fun one. The human chimpanzee. Do we want multiple alignment? Yeah, let's do multiple alignments, except we don't want the overview. That's funny, this was working yesterday. Now let's try this. Oh, this isn't going to let you see this, is it? Well, worked for me yesterday. Here we go. Perfect. So this is a visualization tool that aligns the DNA sequences of two different genomes and shows you how similar the two genomes are at the same parts of the chromosome. So in this case, what we're looking at is the human genome aligned to the mouse genome, and that's why this track reads mouse July 2007. And if we zoom in, now let's zoom out. What's plotted on the x-axis is the nucleotide coordinate. So regions of the chromosomes here up at the top, we see an overall view of the alignment. So right now we're looking at this window of somewhere between 100 megabases and 120 megabases on the chromosome. And we're looking again at comparison of the human and the mouse chromosomes. So the y-axis on this plot is how similar the human and the mouse chromosomes are in sequence at that particular region. So we can see that there's a lot of similarities. So the peaks represent close to 100% sequence similarity between those regions of the chromosome. And if we scan along the chromosome, we can find regions where there is no similarity, like this region right here, between, or very little anyway, between the human and the mouse genomes, and another one there. And the blue bars, if I remember correctly, the blue columns, if that's really blue, maybe it's purple, indicates regions of genes. Those might be exons of genes, for example. And you'll notice that those are always in regions that have pretty good similarity between the human and the mouse DNA sequences. And up above those are shown the positions of the genes. So here, SLC44A1 solute carrier, I believe is that gene name. So there's a gene located there, and there's pretty good sequence identity. And then it's in regions where there aren't genes, like this gap again, where there's no or poor similarity between the human and the mouse sequences, there's no gene there. So that's not surprising. That's an example of degenerating DNA. The DNA doesn't encode a gene or perhaps have a function, so it's free to accumulate mutations that distinguish mice and humans. And it's in regions, again, over here where there are coding regions, genes, that we see high, close to 100% similarity between the human and the mouse chromosome sequences. So that's what a VISTA plot does. It's a simple comparison of, for each position on a chromosome, when you compare two or more chromosome sequences, just plotting on a 0 to 100% scale, how similar are the sequences at each point along the chromosome. And depending on the VISTA plot you look at, the scale of the y-axis might be different. So sometimes it's 100% at the top, and it could be something like 50% where it meets the x-axis. So you do have to pay attention to these sorts of plots for each plot you look at. Now, related to the intellectual basis of this paper, the whole idea is to try to discover how it is that some snakes have lost all of their limbs, and some snakes only have reduced limbs. And so what that means is, and this is maybe the first time we've looked at a phylogenetic tree. This is a classical representation, or a way that evolutionary biologists think about the relationships between different lineages of organisms or species over time. So these tend to run from old time at the top to present time at the bottom. Sometimes these plots will be upside down though. So the point is that the tips of the branches represent extant or currently existing species. So we have presently humans, and boas and pythons are closely related species, so they're represented as a single branch tip, and then there are the advanced snakes, so-called, a group of species, but represented by a single branch tip. And the way to read these is that all snakes and mammals, including humans, had a common ancestor in old time, way back in time. And then as that ancestor proceeded through time there was a point at which the group of animals that became humans, and mammals in general, diverged from the reptilian lineage. So that's what that first branch represents. So then now, at this point, we're talking about the ancestors only to the branch tips that are underneath that trunk, essentially, that branch of the tree. So this is a phylogenetic tree, and that's why we call these branches and tips. So long ago, again, somewhere back in time, there was a mutation or some genetic change that caused that lineage. Right now, we're just talking about reptiles because mammals and maybe other organisms as well are over here, caused limb reduction. That is, why do snakes not have arms and legs today? Well, they sustained mutations that caused their development. So there was limb reduction at some point. We don't know exactly when. This is just a cartoon of the fact that sometime between the divergence of mammals and the divergence of advanced snakes from boas and pythons, there was an initial event that caused their limbs maybe to get smaller. And then there was a common ancestor of all snakes. And boas and pythons diverged, so they separated. They went their own developmental way. And then in a common ancestor to all of whatever we define as advanced snakes, there was a second event that happened that caused them to completely lose all traces of arm and leg growth, limb growth. So what this means is that boas and pythons do actually have reduced limbs, not complete loss of limbs. So if you do find a boa or a python and you take a look at it, you actually can find skeletal evidence that they do still have limbs. They're just really small and we don't actually normally see them, but they're there. So that's the concept behind this paper, is trying to understand the big picture. What are the genetic changes that can cause limb reduction, but not limb loss? So in order to get into this, we should discover or rediscover the terminology about limb axes. So this is pretty straightforward. We've got the upper limb, the lower limb, and then what we would refer to in humans as fingers or toes, depending on whether this is the arm or the leg. And then there are some key genes, proteins, transcription factors in this case, that dictate limb development in many species. And that is that fibroblast growth factor, FGF, tends to be expressed in the upper limb. So that's one of the main genes that controls shoulder to finger development, controlling where the shoulder develops and then how the limb is patterned from the tips of the fingers to the upper arm. So the FGF expression is high towards the shoulder and gets lower towards the tips of the fingers or toes in the case of the leg. The anterior-posterior patterning of the hand has to do with the expression of the protein sonic hedgehog, another transcription factor. And it tends to be expressed in the posterior of the hand. So it's highly expressed on the pinky side of the hand, for example, and not so much on the thumb. And that's how this axis, the anterior-posterior axis of the hand, is patterned. It's a gradient of sonic hedgehog, high to low. Shoulder to fingers is a gradient of FGF, high to low. And then the palm to knuckle or dorsal ventral pattern of the hand is dictated by a Wnt gradient. So Wnt is another transcription factor, another protein that's higher on the knuckle side of the human hand and lower on the palm side. So those three different proteins, and many others, but these are sort of generally speaking, these three proteins have a major role in those three different coordinates of limb development. And in terms of the proximal distal axis, one tissue is really important in specifying the length of limbs or the amount of limb that grows out from the torso. And one of those is called the apical ectodermal ridge. This is a tissue, which I'm going to circle there. It's this ridge that you can see on a developing bud of a limb. So early in development, when the limb is just starting to grow, there's this set of tissues or set of cells called the AER. And their identity is induced by cells that are just beneath them, called mesenchymal cells. And this is a feed-forward mechanism where those mesenchymal cells tell the apical ectodermal ridge that it's an apical ectodermal ridge, and then the AER supplies transcription factors or signals that enable transcription factors in the mesenchyme to replicate, which is what mitotic state means. So this is a feed-forward mechanism. The AER tells the tissue underneath it to keep dividing, and that's what causes the limb to grow out from the torso. And then the mesenchymal cells stimulate the AER. And of course, at some point, this has to stop because that's how limb growth stops. If this process happened forever, then of course, the limb would keep growing and growing and growing. The AER is both necessary and sufficient for limb development. So here's some quick information about this. Necessity, that's the lose it type experiment. So what developmental biologists did was they'd actually remove the cells of the AER, and that causes limb development to stop. So if you don't have the AER, your limb doesn't keep growing. So it's necessary for limb growth. Sufficiency, the move it type experiment. You can take the AER and put it somewhere else, and you can discover that you can cause the renewed growth of a limb by moving it somewhere else. So for example, in this example, they can take the AER and put it over a place in the body that isn't normally going to develop a limb, and moving the AER on top of that tissue is enough to make that tissue start to grow a limb. I mean, imagine you did this to the chest of an organism. Maybe you could make it grow, start growing a limb out of the middle of the chest where it wouldn't normally. So that dictates the sufficiency. If you move this AER somewhere else, that's all you need to do to stimulate the beginning of limb growth. And so this is a cartoon of that feed-forward mechanism that I mentioned. So in the early tissue, there are regions of the body that have concentrations of FGF signaling and not others. So not in the middle, for example. So this might be where arms are going to develop, and this is where legs are going to develop. So FGF10, fibroblast growth factor 10 expression, is restricted to particular parts of the early embryo. And in those regions, those FGF10 stimulate in the AER, this triangle I'm representing it as. FGF causes the expression of Wnt, which is another transcription factor that's a signaling protein that causes the expression of other proteins like fibroblast growth factor 8 in this case. And FGF8 sends signals to that mesenchyme tissue that's just underneath the AER, and that causes those cells to produce more of the same protein, FGF10. FGF10 causes Wnt expression, causes FGF8 expression, causes more expression of FGF10, and the cycle continues. And FGF8 not only keeps FGF10 being expressed and telling the AER to stay AER, but it also then signals the mesenchymal tissue, the tissue just underneath the AER, to keep replicating, and that's what causes limb growth. The AER keeps telling the cells just next to it in the limb, hey, keep dividing, we're growing a limb, so you need to keep growing, keep dividing and producing more cells. There's another part of limb development that's again, the AP axis, the thumb to pinky axis. And this has to do with a set of tissues like the AER, except this is called the ZPA, the zone of polarizing activity. And that is a set of tissues at the posterior part, so like the pinky part of a limb in this case, or a hand, that defines the posterior. And so you can do things like move that set of tissues, and whatever signals they produce, which is the critical part here, these tissues are probably producing hormones or paracrine signals or other signals that are telling the cells around them what to do. So these are kind of like boss cells. And if you move those cells into another organism, say at the anterior part of a limb, so they have the normal ZPA, which would normally produce pinky, and you have a new ZPA, what that does is it winds up confusing the tissues because it thinks, well, I'm supposed to be pinky down here, oh, but I'm supposed to be pinky up here, and you can actually produce organisms that have limbs where the hands essentially are confused. They have two pinky-like sets of digits with sort of middle digits in between. And one of the key molecular players in the zone of polarizing activity is sonic hedgehog, or SHH. It is a gene that's produced in that posterior of the limb buds. So it's important in defining the ZPA. So you can do things like not just move the ZPA, but you can actually synthesize sonic hedgehog, the protein, and just put it in a limb bud, and you get the exact same sort of weird patterning that we just saw. So it's not just the cells, that you can actually look at the specific effects of particular proteins and show that they are mainly responsible for the effects that we see. Apoptosis is a set of two Greek terms. So there's the prefix apo, and then there's the tosis. So you can pronounce this however you want. I always thought it was pronounced apoptosis, and it sounds cool because what apoptosis does is it tells cells to die. So apoptosis is referred to as programmed cell death. Some of the cells that are produced during development aren't meant to survive until the organism is an adult. Some cells are developed or produced and then actually meant to die later. So apoptosis plays a critical role in development. It's also playing an important role in things like cancer progression. So some cells have built-in triggers that if something goes wrong in the cell, it should basically commit cellular suicide, apoptosis, and kill itself or die to protect the greater organism. So apoptosis, or apoptosis, you can call it either, is programmed cell death. And this is also important in digit formation because that's how humans don't have webbing between our fingers and toes, but ducks do, for example. This involves another set of proteins called bone morphogenetic proteins, or BMPs. And so we can take a look at how this works in both chickens and ducks and compare. And what those BMPs do is they signal regions of tissues that are going to undergo apoptosis. So we would expect BMPs to be expressed in humans in between the digits, but not in, say, ducks. And BMP then is involved in destroying or removing the apoptosis of the tissue in between the digits. Then BMPs have inhibitors like gremlin. So we would normally draw that as a repressive interaction, where gremlin, if the protein gremlin is produced in the tissue, it will stop BMP from apoptosis. So what we expect then is that in organisms that keep their webbing, you would have BMP expressed here normally, like in a chicken, and that would cause that webbing to disappear. Those cells would undergo apoptosis and be destroyed. But in ducks, you see the presence of gremlin. So the dark spots here with the arrowheads indicate regions where the gremlin transcript and probably the gremlin protein is being produced. And in those tissues, it is preventing BMP from causing apoptosis. Thus, the duck maintains the webbing where there's no gremlin expression in chickens. And so BMP is ready to do its job and able to do its job, causing the apoptosis of those tissues. Now, there are a few experimental approaches that we should talk about. One is in situ hybridization. So if you have a developing organism, which in this case is going to look like a platypus with a tail and a hind limb and a forelimb bud, and there's its head, maybe. Sure, why not? If you want to know where in the body a particular gene is being transcribed, one of the classic methods is an in situ hybridization. And the concept here, and we saw this in some of the slides just now, for example, the zone of polarizing activity, we saw dark spots where the ZPA is located. And we learned that that had something to do with the sonic hedgehog protein. So if we want to know where sonic hedgehog is expressed in an organism, the first thing you do is you collect an organism. You have to kill it, unfortunately. This is often done in embryonic tissue for developmental biology research. And you fix the tissues, which basically means you permeabilize them to stains. And in this case, what we're going to do is we're going to take, or the authors anyway, would take sonic hedgehog messenger RNA complement. So in other words, if we want to know where in the organism sonic hedgehog messenger RNA is expressed, which is represented by these dark spots, those are regions where SHH transcript exists, that transcript has a particular sequence that's specific to SHH, sonic hedgehog. So in this case, I'm just going to make up, except we don't have T's in transcripts, we have U's instead of T's. So I'm just going to make up a short sequence, AUG, UGC, A, U. So that's the sequence of the messenger RNA. The hybridization part comes from generating what's called a probe, which is antisense transcript. And what I mean by antisense is it's complementary in sequence to the messenger RNA. In other words, where there's an A in the messenger RNA, there's a U in the probe RNA. So this is a single-stranded RNA molecule that's complementary to the target, which is the gene we're interested in studying. So once we have this synthetic probe, single-stranded RNA sequence, it's also complexed with sometimes enzymes or fluorescent molecules. In this case, we'll talk about fluorescent molecules, but that's not always the case. So it may be, for example, that all of the A's are coupled to a fluorescent molecule. So then the in situ hybridization part is take a pool, millions of copies of the same probe sequence that's labeled with fluorescent molecules, and you basically bathe the fixed embryo in this probe. And anywhere there is a messenger RNA sequence, anywhere in the organism's body that's complementary to this probe, the probe sequence will base pair like drawn here. And so what that means is that this probe sequence will accumulate only in regions of the tissues where they have a messenger RNA sequence they would bind to. And that shows us where that messenger RNA sequence exists in the body of the embryo. So that's in situ hybridization. Finally, there are a few components of how an enhancer can be identified and defined. And that's one of the really big questions in a lot of developmental genetics research is enhancers, as we saw earlier, don't have to be really close to the transcript or the gene. They could be really far away. They could be hundreds or thousands or millions of base pairs away from the genes they regulate. So it can be tricky to figure out what parts of the chromosomes are enhancers for which genes, because there could be lots of genes around physically in the vicinity of an enhancer, but that enhancer might not work on any of those genes. It might work on a gene, influence the transcription of a gene that's millions of base pairs away. So there are a few things that scientists look for when they're trying to identify enhancer sequences among the other millions and millions and millions of base pairs of DNA. One is they use those VISTA plots to seek conservation. So enhancer sequences, because they have functional roles like genes do, we predict that enhancer sequences will be highly conserved between even very different species, just like genes are. The other things that we expect are that enhancer sequences, as opposed to sequences that don't have any role in transcription, enhancer sequences should cause the expression of a reporter gene. And this can be done, in this case, in two different ways. There are many others. In vivo, in the organism, which is what in vivo stands for, inside a living organism, a LacZ assay is one way to check whether or not a putative enhancer, a potential enhancer, actually has enhancer activity. In vitro, or in glass, like in a test tube, you can also perform a Luciferase assay to essentially ask the same question. Does a particular DNA sequence have the function or the ability to drive expression of another gene? And the third thing is that an enhancer sequence should actually be bound by a transcription factor, and we can test that. So here we go, last few points. The beta-galactosidase or LacZ assay, the idea here is we have a reporter gene. So here's a chromosome, here's the LacZ gene, which produces a protein called beta-galactosidase. It's an enzyme that cleaves a particular sugar, and it can also cleave a particular substrate to produce a color. So this is a color-based assay. And the idea is anywhere in the body of an organism that LacZ transcript is produced, you get beta-galactosidase protein. And then wherever in the body of the organism that enzyme is produced, when you bathe that, just like the in situ hybridization, if you bathe the, say, embryo, fixed embryo, in the substrate, then only the regions of the body that contain this enzyme will catalyze the enzymatic change of that substrate to a colored product that we can see with our eyes. And then we can see where in the organism this enzyme is being produced, beta-galactosidase. So the functional part of that is that you have to have an enhancer. So what you do is, what the scientists do is, you put putative enhancers, potential enhancer sequences, upstream of LacZ, and then you ask, does that actually produce LacZ transcription? If the enhancer is an enhancer, you see LacZ transcript, and those get translated into the beta-galactosidase enzyme, and then those enzymes react with the substrates that are provided to produce like a dark color in that region of the organism. And you can measure both where the LacZ is produced and also how much is produced to get a sense of how good or whether or not an enhancer, potential enhancer, is good at all. You can do the same thing, but using an enzyme called Luciferase. The concept is exactly the same. It's just that Luciferase is not used in the context of looking inside a living organism or a fixed embryo, as in the beta-galactosidase case. These are usually performed in vitro or in a test tube, where you have a piece of DNA, same story, the potential enhancer cloned or placed upstream of another reporter gene, in this case, the luciferase gene, which produces, like beta-galactosidase, an enzyme called luciferase, which catalyzes the reaction of a substrate, but instead of producing some sort of pigment that we can see, the luciferase assay produces light. So in the test tube, you can combine the DNA construct that contains a potential enhancer with luciferase. You throw in all of the things that are needed to produce protein from this. So you do in vitro transcription and translation to produce protein. And if any protein is produced, luciferase protein that is in this case, that's evidence that this enhancer may be functioning as an enhancer. And if it's a really good enhancer, we'll get a lot of luciferase protein, we'll get a lot of light in the test tubes that can be measured in the laboratory. Finally, the enhancer, if it's an enhancer, should be able to be bound by a transcription factor protein. The concept of the electrophoretic mobility shift assay is based on gel electrophoresis. If you're not familiar with this, this is the idea that you can load, in this case, nucleic acids, like RNA or DNA, into holes that are in a gel of some sort of matrix, like Jell-O. And if you apply an electric field, because nucleic acids are negatively charged, they will migrate towards the positive end of the gel if you place an electric field across the gel. So nucleic acids go in the top, and then they are pulled through the gel, and they're separated then based on their physical size. So small pieces of nucleic acid, because they're small, they can migrate their way through the matrix of the gel more easily than large pieces of nucleic acid. So the small pieces of nucleic acid go all the way down to the end of the gel. And what would happen if this nucleic acid happens to be a potential enhancer sequence? Well, if it's an enhancer, and if, let's say, it's bound by generic transcription factor, TFX, transcription factor X, if TFX binds this enhancer sequence, this short piece of nucleic acid. Let's say this is 50 base pairs long. You've got a double-stranded piece of 50 base pairs of DNA we think is an enhancer or contains an enhancer. Well, what we do to find out if it binds transcription factor X is we premix, say in a test tube, that nucleic acid, that 50 nucleotide fragment, with some of the proteins. We'll call transcription factor X this black blob. So we premix those in a test tube, and then we load that whole mixture into an adjacent well on a gel. If the blue nucleotide is an enhancer, and if it binds transcription factor X, what will happen when we look at where this nucleic acid winds up on this gel? If the blue nucleic acid is actually physically bound by a transcription factor, that makes that protein nucleic acid complex relatively large compared to the nucleic acid by itself. So what that will do is it will retard its migration through the gel. In other words, that nucleic acid together with the protein is bigger. So it doesn't go through the gel as quickly as the DNA by itself. That is the mobility shift. So if the DNA, the potential enhancer, binds a protein that we mix in with it, then that will cause that DNA band to show up higher on the gel. It will look like a larger DNA product than it actually is, because these two are actually the same size piece of DNA. The only difference here is that in this lane, we've mixed the DNA with the protein. Last question, what would happen then if we mutated that blue DNA sequence so that a critical part, and here's double helix now, I'm showing this as a double helix instead of a single line representing a double helix. What would happen if we mutated a really important part of that enhancer sequence where that protein would bind? What happens if we mix that together with our proteins? So we have proteins in a tube, and then we have these mutant versions. Oh, that looks like a, well, I thought that looked like a face or something. Maybe it does. So we have these mutant enhancer sequences that the protein no longer recognizes because we've changed its binding sequence. What happens now when we load that on the gel? The only thing we've done now, again, between the second lane and the third lane is we've changed a small portion of that DNA sequence. And once again, we might see that the DNA sequence no longer binds the protein. So these are two different complementary experiments. The one in the second lane and the third lane are complementary experiments that together suggest not only that this is a sequence that's bound by this transcription factor, but that the specific sequence of that nucleic acid is important for the binding. The reason that this could be important is because transcription factor X could instead be a general DNA binding protein, BP. In other words, what if the protein we mixed in with the DNA that we think is an enhancer, what if that protein doesn't recognize specific DNA sequences? What if it just recognizes DNA and binds it, right? Then that wouldn't be good proof that that protein is specifically recognizing the DNA sequence. And that's important for an enhancer. It has to be specifically recognized by the transcription factor. And transcription factors have DNA binding domains. So this is, again, an important complementary experiment comparing the sort of wild type enhancer sequence and its protein binding ability with a mutant to show that you can abolish or get rid of that specific interaction, if there is one, between the protein and the potential enhancer.